Hello, everyone. Thank you for attending my talk this morning. My name is Per Asfer Ad Astra, and I'm a clinical metagenomics fellow in Texas Children's Microbiome Center here in Houston. Today, I'll be discussing how we're using long-read sequencing to generate case-specific reference genomes for pathogen strain typing. Specifically, I'll be discussing a case where Candida was identified in a mother and neonate following fetal surgery. Our case starts at 20 weeks gestation with the discovery of a fetal neurological tube defect, spina bifida. At 25 weeks, the mother and fetus successfully underwent surgery at Texas Children's to correct this defect. However, shortly thereafter, the mother began experiencing significant abdominal pain, went into preterm labor, and delivered at only 27 weeks. Now, 3 days post-delivery, budding yeasts were identified in a blood culture taken from the neonate immediately after delivery. Now, if you're not, and specifically, Candida parapsilosis was what was identified. Now, if you're not familiar, Candida parapsilosis, or C. parapsilosis, is a leading cause of non-albican candidiasis and considered a priority pathogen by the WHO. Primarily found on the skin, infections are often linked to nosocomial spread, and those at highest risk for infection being patients in the ICU or, as in our case, preterm neonates. However, budding yeasts were also then later identified in the mother's placental tissue. Therefore, as part of a retrospective study, physicians involved in the case wanted to determine if the budding yeast identified in the mother and the neonate were the same species and strain of Candida. In order to answer this question, we used long-read sequencing to generate a C. parapsilosis reference specific to this case. To do so, DNA was extracted from the neonate blood culture and sequenced using Nanopore's latest library and flow cell chemistry, V14 and R10 respectively. Raw data were then translated into FASTQs using Dorado, assembled with Fly, and polished with Medaka. The resulting assembly consists, depicted in this circus plot here on the left, consists of 8 contiguous nuclear chromosomes and 1 contiguous mitochondrial chromosome spanning 13 megabases. Benchmarking of universal single-copy orthologs specific to fungi estimates the completeness of our assembly at greater than 96%, on par with the NCBI reference assembly for this species. Next, using an Illumina-MiSeq, we generated short reads from both the neonate blood culture as well as the mother's placental tissue. These reads were then mapped to the reference we generated, and the average nucleotide identity of each library calculated relative to this reference and visualized in the form of read recruitment plots. For reference, isolates of the same species will have an average nucleotide identity of greater than 95%. Here we can see the read recruitment plots for the neonate blood culture on the left, with an average nucleotide identity of 99.7%, and the placental tissue with an average nucleotide identity of 98.5%. Based on the lower of these two values, we can say that the Candida in both the mother and the neonate are 98.5% identical relative to the reference we mapped to. Therefore, returning to our original question, are these budding yeast or Candida the same? Our current data supports that these Candida are the same species, C. parapsilosis. However, these data are insufficient to determine if they are in fact the same strain. Moving forward, we're currently collecting additional data to improve the base accuracy of our assembly, as well as look into some potential strain-specific structural variation that we've seen, both of which are necessary for performing functional and phylogenetic analyses that we will be including in a publication of this long read-only assembly of an important human pathogen. And with that, I would like to thank and acknowledge my advisors, Ruth Ann and Jennifer, and I'm happy to take questions here or at my poster. Thank you.